Hi, class. In this recording, we're going to focus on neurotransmitters, what they are, how they work, and different kinds of neurotransmitters. So let's give you a little historical context. Neurotransmitters were discovered back in 1921. Oop, here we go. Back in 1921. And while these neurotransmitters were discovered, they were found to be we found that neurons were releasing chemicals to the chemical synapse. The chemical synapse is going to have a small gap, and that gap is referred to as the synaptic cleft. The gap was discovered by Raman E. Kajal, and then the neurotransmitters themselves were discovered by Otto Loewe. Otto Loewe did some very landmark experiments where he took frog hearts and flooded them with salt water or saline. He stimulated the vagus nerve of the first frog, heart slowed down, and then he removed the saline and then found it slowed the heart of a second frog. In other words, that liquid that was around the heart in the first frog must have carried some kind of a chemical from the vagus nerve to the heart of the second frog. And he named this substance the vagostuff, or vagus substance, because it came from the vagus nerve. And it was later renamed to be acetylcholine based off of its chemical composition. Acetylcholine was the first discovered neurotransmitter. It's also the most common neurotransmitter in the human body. So as we're looking at our electrical synapses, neurotransmitters don't exist in electrical synapses or within our nervous system, generally speaking. They do exist. I misspoke there. They do exist. Electrical synapses do exist in our nervous system. The most common instance of an electrical synapse that we talk about in this class is the intercalated discs in between heart muscle cells, or in between cardiomyocytes. We have a few electrical synapses between neurons and neuroglia, and we spend a lot of time talking about the electrical synapses in the cardiac muscle cells. And occasionally, if we have time, we get to it in the smooth muscle cells. All electrical synapses rely on gap junctions. You can think of a gap junction as a hole between the first cell and the second cell. And this hole directly connects the cytoplasm of these two cells. And if the cytoplasm can flow from one cell to the next cell, ions within that cytoplasm can flow, propagating the electrical signal. The big advantage of electrical synapses is they skip the neurotransmitters entirely, so we have a very quick signal transmission. There's no delay related to those neurotransmitters. Now, there's a downside of the electrical signals in that there's no integration, there's no processing of information. It's just, okay, got it, go, as opposed to, okay, I've got this information, I've got that information, what should my response be? We can't integrate excitatory and inhibitory signals with our electrical synapses. So as we're looking at a chemical synapse, which we'll spend most of our time focusing on, we have the presynaptic neuron with its axon terminal, and at the bulb at the end, there will be neurotransmitters released. Those neurotransmitters are in synaptic vesicles. And there's always going to be a pool of synaptic vesicles within the bulb or axon terminal that can be brought to bear. They're kind of on deck or in the hole, so to speak, ready to be exocytized. That postsynaptic neuron, which is shown at the bottom of the screen here, is going to have proteins on its membrane that receive the neurotransmitters. These are going to be ligand-regulated ion gates, or you can think of them as chemically gated ion channels. That's another way to talk about them. Here's a photomicrograph of this process. We can see here we have an axon, here's an axon terminal, and then here's the neurosoma of a postsynaptic neuron. So this one neurosoma, it looks like it has a couple dozen axon bulbs synapsing with it all at the same time. That's pretty amazing. Now, as we're looking at neurotransmitters, neurotransmitters are thought of as just chemicals or molecules that are released from the presynaptic neuron and then bind to the postsynaptic cell and alter its physiology or alter the cell's chemistry. In our bodies, we have over 100 neurotransmitters. We're not going to talk about all 100 plus neurotransmitters have been identified. We're going to focus on the broad categories, the 4 broad categories: acetylcholine, amino acids, monoamines, and neuropeptides. So let's talk about acetylcholine-based neurotransmitters. Acetylcholine-based neurotransmitters are, well, just based on acetylcholine. It's so unbelievably common that acetylcholine, this one neurotransmitter, is a class by itself. And as we're looking at this acetylcholine, we can see here that we have, if we cleave it right here, we have an acetyl group, and here we have a choline group. So hence, that's its namesake, acetylcholine. It's formed from the acetic acid and choline being bound together. We can also have amino acid-based neurotransmitters. And as we're looking at these amino acid-based neurotransmitters, they're, well, based off of amino acids. They include glycine, glutamate, aspartate, and gamma-aminobutyric acid. Gamma-aminobutyric acid is a mouthful, so a lot of times people will say GABA instead. We also have monoamine-based neurotransmitters. Monoamines, as their name implies, have a mono what? Only one amino group. They're going to be synthesized from an amino acid, but the carboxylic acid functional group of the amino acid is removed while we retain the amino group. Common monoamines are going to include the catecholamines that we talked about with when we had our discussion on the endocrine system. We had epinephrine, norepinephrine, and dopamine. Dopamine was one that we did not talk about in detail with the endocrine system. And we also are going to include some other monoamines that are not going to be associated with the sympathetic nervous system. These include histamine, ATP, and serotonin. We're also going to have another category of neurotransmitters known as neuropeptides. Peptides are you could think of a peptide as a small group of amino acids, typically 2 to 40 amino acids. Sometimes this is referred to as an oligomer of amino acids as opposed to a polymer of amino acids. And the neuro prefix implies that they're going to be active or actively affecting neurons. We are going to have our neuropeptides stored in secretory granules within the cells, and they include some pretty common ones such as cholecytokinin and substance P. These neuropeptides, those 2 neuropeptides, are going to be very important for our digestive system. We'll talk about those in more detail in Bio 314 next semester. Some are going to function as hormones or neurotransmitters. Some can be released from the digestive tract, like those 2. These gut-brain peptides can cause food cravings. They can limit food cravings. When we think of these neuropeptides, they are multi-purpose signals that can act as either a hormone, so they travel through the bloodstream, or a neuromodulator, in that they regulate the activity of a neuron. Some of our neurotransmitters are gaseous neurotransmitters. The single most talked about gaseous neurotransmitter is nitric oxide. Carbon monoxide, to a lesser degree, is also focused on as a gaseous neurotransmitter. This nitric oxide is very widely talked about because it causes the relaxation of smooth muscle cells within the blood vessels. It lowers our blood pressure by dilating our blood vessels. Now, these gases are small. They can travel through membranes very quickly. And because they travel through membranes very quickly, we cannot store them. Instead, when we need them, we synthesize them on the spot in our synaptic vesicles, and they move in and out of the cell membranes with impunity, rapidly going to the target tissue, that postsynaptic cell. That's all we have for this recording on neurotransmitters. If you have any questions or comments, please feel free to post them on the class discussion board or shoot me an email. And as always, happy studies.